Hi, good morning, everyone. This is Leah with Headwaters Science Institute. Excited to welcome you to another Lunch with a Scientist talk, where today you're going to get to know Jessica Tse Barra. She's a microbiologist, and she's going to talk to you a little bit about mechanisms of antibiotic survival in bacteria. So that should be a really interesting talk. But before I call her in to talk to you, I wanted to give a little bit of updates from Headwaters. So Jessica's talk is a part of this Women in STEM speaker series that we've been doing over the last week and a half or so. She is the fourth in a series of 5. Tomorrow night, we welcome Tucker Malarkey, fabulous author of national best-selling books, to talk about her newest book, Stronghold, which is a story of a man's quest to preserve the life of wild salmon. And so please tune in at 5:30 tomorrow. It's going to be a really interesting talk. We created this series to celebrate online learning and to celebrate how far we've come last spring and this summer with all of our free online learning materials, and to look towards this fall. We're hoping that you'll see what we're doing and see the power of technology in bringing free online learning to kids and be inspired to donate. So you can join us at headwatersscienceinstitute.org/donate. If you like what we're doing, you can find free online learning resources there that will keep your kids engaged in hands-on science. You can find talks like these and a lot more content. So in rounding out our celebration for these 2 weeks, I just want to remind everyone we're doing a special discount on our online science camp this fall through Friday. You can save $100 on your registration. If you are a high school student wanting a high-level research opportunity, please check out our website for that. We are also running a yearly raffle where you can win some great prizes. Again, check out our web store on our website to support our programs and participate in our raffle. And then lastly, for little kids, you can join in our scavenger hunt. We put a video out about it last Friday, and you've got until this Friday to go out there and find some of the things on our scavenger hunt card, and you might win a prize. So you can find out more about that on our website. Again, we're just so thankful for your support of our online learning programs. And we're about to have another great talk today. I am pleased to welcome Jessica Tse Barra. Hi, Jessica.
Hi. Thank you for having me, Leah.
You're very welcome. So excited to hear your research. Another powerful woman in science. So we're very excited. And where are we talking to you from today?
Oh, I'm in Massachusetts.
In Massachusetts. So we're in California here. It's morning for us, and it's a little bit after lunch for Jessica.
Yep.
So I am proud to introduce Jessica. She graduated from Wellesley College in Massachusetts. She studied biological chemistry and did undergraduate research on cyanobacteria and the stress response. She later received her PhD in microbiology at the University of Wisconsin-Madison, studying how bacteria survive antibiotic stress. And she currently works as a scientist at a small biotech company. So she's going to tell us all about antibiotics today, and I will turn it over to you.
Great. Thank you, Leah, for the kind introduction. As Leah said, my name is Jessica Tse Barra, and today I'll be telling you about mechanisms of antibiotic survival in bacteria. So this is my talk outline. First, I'll talk about discovery of antibiotics, followed by the emergence of antibiotic resistance. I'll talk about how antibiotics work and mechanisms of antibiotic resistance. Next, I'll talk about persistence, which is the topic of my PhD research, along with the role of a small molecule called ppGpp. And finally, I'll talk about the future of antibiotic therapy, as well as my path to science and my life as a microbiologist. So I'm sure many of you have encountered antibiotics during your lifetime. Antibiotics are compounds secreted by bacteria or fungi that can kill or inhibit other microorganisms. The antibiotic penicillin was actually first discovered by Alexander Fleming in 1928. What you see here is an image of an agar plate containing nutrients for bacteria. Initially, Fleming was studying Staphylococcus aureus, which is an organism known to cause skin infections. So what he did was plate Staph aureus onto this agar plate and left the plate hanging out on the bench for some time. What he later discovered was there was actually a mold growing on top of his plate, which was a penicillium colony. And what you see around the mold is something that we call the zone of inhibition, where there is no growth of any of the bacteria that he plated. Further away, you see that there is growth of normal-sized Staph aureus colonies. So what later was discovered was that this penicillium mold was secreting a compound called penicillin that helped to inhibit bacterial growth. With its isolation and manufacturing, penicillin ended up saving a lot of people, and people no longer died from simple scrapes and cuts. However, people thought that antibiotics were a cure-all and that bacterial infections were a thing of the past. With its frequent use and misuse, antibiotic resistance actually started arising. So what you see here is an image of a timeline dating from 1930 to 2005. And above it, you see the year in which a specific antibiotic started being used. So for example, chloramphenicol started being used in the late 1940s. Below this timeline, you see the year in which antibiotic resistance was first observed. So for chloramphenicol, you see that in the late 1950s was when resistance started being observed in the field. So how did these antibiotic resistances start to arise? In order to talk about that, we need to first talk about how antibiotics work. And antibiotics target cellular processes in bacteria. So this image is an image of a typical bacterium. There are many different types and species of bacteria, but this image highlights many common features that bacteria have. So here you see the capsule, cell wall, and plasma membrane, all of which provide a protective coating for bacteria. You see ribosomes here, which are depicted by these two blue circles, which help the bacteria to make more protein. And then finally, in the center of the cell here, you see this mass of red, and it's the nucleoid, and it houses the DNA. DNA contains the genetic information for bacteria to grow, live, and make more of itself. So let's start by talking about the cell wall targeting antibiotics. Again, the cell wall forms a protective coating around the cell, highlighted here. So examples of cell wall targeting antibiotics include beta-lactams. And so an image shown here is an example of a beta-lactam, and highlighted in red is the beta-lactam ring, which will become important later. So examples of beta-lactams include penicillin and methicillin. Another cell wall targeting antibiotic is vancomycin. So beta-lactams work by preventing the protein used to link pieces of the cell wall together from working. So down here, I provide an example. You see that there's one piece of cell wall up here with a pentapeptide chain, and then you see a second piece of cell wall below with corresponding pentapeptide chains. Normally, how the cell wall is constructed is that this protein will bind these two chains and link them together. However, with the introduction of the beta-lactam antibiotic, it binds to this protein, preventing this from occurring and preventing the linkage from occurring. Next, we'll talk about ribosome targeting antibiotics. Again, the ribosome links together amino acids to make proteins for the cell. Examples of ribosome targeting antibiotics include tetracycline, chloramphenicol, and erythromycin. And how chloramphenicol works is that it prevents the peptide chain, which is a chain of amino acids to make a protein, from elongating. So what you see here is an image of a ribosome. The orange subunit subunits you see here are the ribosome, and the blue squigglies you see here are transfer RNA that contain amino acids. So what's happening here is that this middle transfer RNA has a chain of peptides, amino acids to form a peptide chain, and to make new proteins, new transfer RNA containing amino acids get transferred into the ribosome and then get added into this peptide chain. So when chloramphenicol binds, it binds between these two locations, preventing this transfer from occurring. And this would prevent the bacteria from making more or adding more amino acids to this chain. DNA synthesis, now we'll talk about DNA synthesis targeting antibiotics. DNA holds the genetic information that bacteria need to grow, live, and make more of itself. What's also important to know is that DNA needs to unwind in order to duplicate and express genes, among other things. So again, the DNA is located in the center of the cell in the nucleoid. Examples of DNA targeting antibiotics include ciprofloxacin and norfloxacin. And how these work is that it prevents the proteins used for unwinding the DNA from working and prevents cell division. So this is an image of DNA. These are the proteins that are used to unwind the DNA. And so ciprofloxacin would bind and prevent this from happening, prevent unwinding of the DNA from happening. So how do bacteria develop resistance to antibiotics? Here I'll go over several examples, but there are many examples out there. So antibiotic resistance arises by acquiring foreign DNA from plasmids. And plasmids are small circular pieces of DNA that have genes that can confer resistance. For example, resistance plasmids can contain beta-lactamases, which are enzymes that can degrade beta-lactams. So shown here is a typical bacterium with its nucleoid and DNA. And what you see in yellow is a plasmid containing beta-lactam. So normally, as I've discussed before, the beta-lactam can prevent this protein from linking together pieces of the cell wall. However, if the cell has a plasmid that contains a beta-lactamase, the beta-lactamase can degrade this beta-lactam ring, thus removing its ability to bind to the protein. And then the protein can do its job and link the pieces of cell wall together. So then the cell can go along its merry way and continue to grow. Next, antibiotic resistances can also arise by mutations. Mutations can occur in the ribosome that can prevent chloramphenicol from binding. So again, this is the bacterium with its DNA in the center. Mutation has occurred, depicted by the star here, and the mutation has occurred in the ribosomal RNA, which makes up the ribosome. So a mutation, corresponding mutation on the ribosome is here, and it prevents the chloramphenicol from binding. So then this allows this amino acid to get added into this elongating peptide chain. So ribosomes can continue to make proteins. Finally, antibiotic resistance can also occur when bacteria produce more antibiotic efflux pumps to pump out the antibiotic. So here is our lovely bacterium, and it has expressed several efflux pumps depicted in yellow here. So how these work is that it pumps out the antibiotic that's in the cell. So this one gets pumped out, and then this one gets pumped out, and it gets pumped out of the cell, effectively lowering the concentration of the antibiotic in the cell. And so with the decrease of the antibiotic in the cell, the DNA can now get duplicated and transferred into new cells. So how do you measure antibiotic resistance? Oftentimes, resistant bacteria can grow in the presence of the antibiotic, and growth is characterized by cloudy bacteria cultures or lawns of bacteria. So what you see here is an example of two agar plates which contain nutrients for bacteria. And in these discs are various concentrations of antibiotic or different types of antibiotic. And so how this assay works is that a high concentration of bacteria is plated onto the agar plates. The antibiotics are within these discs are put onto the agar, and then the antibiotic is diffused throughout the agar. These plates are incubated so that the bacteria can grow, and after the bacteria is grown, you can actually see whether the bacterium is sensitive to the antibiotic or not. So if you remember from one of the first slides, we saw something called a zone of inhibition. So here, this antibiotic and this antibiotic gets secreted, and the bacterium is sensitive to it because there's an area in which they cannot grow. Whereas if you look at these three antibiotics at the bottom here, the bacteria can grow around it just fine, indicating that it's actually resistant to the antibiotic in these three discs. Another way to determine antibiotic resistance is through a minimum inhibitory concentration, or MIC assay. And for resistant strains, the MIC actually increases. So what you see here are several wells containing media or broth that'll grow the cells, as well as some cells. So what you see here is a well with blank media or nutrients. So it doesn't have any cells in it, because you need to make sure that your media is not contaminated to begin with. So this is your negative control in this assay. In this well here is bacteria and the media without any antibiotic. So this is your positive control. So it makes sure that your bacteria is growing properly without the presence of the antibiotic. And from this well onward are increasing concentrations of antibiotic and cells. So you see that the couple of concentrations are low, and the bacteria can grow in the presence of it. The first concentration at which there is no growth is called the MIC, the minimum inhibitory concentration, and everything after that shouldn't have any growth either, because that concentration of antibiotic is actually higher than the MIC. So if a bacterial strain is resistant, what you'll see is that they'll grow several times higher in media containing antibiotic that's several times higher than the MIC. Another way to quantify bacteria is actually quantifying its survival after antibiotic treatment. So what you see here are a couple of flasks where the cells are just growing in media or broth, nutrient media or broth. And so as they grow more and more, the media grows from clear to cloudy. And so at various time points, what you can do is take dilutions and plate the dilutions onto agar plates and then incubate them so that the bacteria can grow on top of the agar. And you can count these circles or colonies here called colony forming units. And that's how you're able to quantify how much bacteria is in your flask at a given period of time. At the same time, you can also add treatment to your cells. So here, indicated by the blue, we've added antibiotic. And similarly, you can perform dilutions and plate on agar over time and incubate them so that you can get a count of how many bacteria are in the flask at specific time points. And what you can do is determine the number of survivors at each time point and then graph them here. So if you're comparing a specific time point at time 0, you get a certain number of colony forming units present on your agar. And then over a period of time, you get different numbers of survivors or number of colonies that you can then compare to your time 0 to see if the cells are growing or dying over time. just to reiterate the point here, we can demonstrate a couple of these in this next couple of slides here. So to demonstrate growth, you see that the bacteria here in red are in the presence of media or broth highlighted in this green box here. And so if the cells are growing, over time, you'll see that the number of colonies actually increases past the 100%. And that demonstrates that the bacteria are actually growing over time. If you grow the bacteria in the presence of an antibiotic, which is highlighted, depicted by this blue box here, and the bacteria are resistant, then the bacteria can either degrade the antibiotic or pump it out. And so that will show that the bacteria can actually grow in the presence of the antibiotic over time here. Growth. And then finally, we'll talk about persistence, which was actually the topic of my PhD thesis. And persistence, in persistence, the bacteria is grown in the presence of an antibiotic, again depicted by this blue rectangle here. And in persistence, what happens is that the cells actually quickly die in the first couple of hours or so, followed by a plateau where the cells neither really grow nor die while in the presence of the antibiotic. And this type of curve here is called a biphasic kill curve, where we see in the first phase, we see rapid decrease in the number of cells. And in the second phase, we see that the number of cells actually plateau over time. And recently, there's been a lot of research on persistence. However, this phenomenon is actually not new. It was first discovered and published by Dr. Joseph Bigger back in 1944 when he was treating staphylococcal infections with penicillin. And he summarized it really well here. So these cells that survive in the plateau are called persisters. And how he summarized it was as follows. The only hypothesis which seemed to explain the occurrence of a small number of survivors, or persisters, out of the millions of cocci originally present is that these differ from the majority of their fellows in that they are capable of surviving a concentration of penicillin which, in the time of action allowed, kills the others. So these persisters can survive the penicillin while the others cannot. And so for my PhD work, I worked on an organism called Bacillus subtilis. It is a soil-dwelling bacterium that is known for its ability to sporulate and form biofilms. So in this really cool micrograph here, you can see in green are actually the spores. And for biofilms, you can see that a colony looks like this, really cool looking. And then if a culture is left in liquid undisturbed over time, you can actually see a biofilm form on top of the liquid. What's also really cool about it is that Bacillus subtilis is a model for studying many other pathogens that are similar to it in the same phylum as it. This includes Staphylococcus aureus, which we've already discussed, Enterococcus faecalis, and Listeria monocytogenes, the foodborne pathogen that you might know about. And how we can use it is because Bacillus subtilis has many conserved pathways and regulatory mechanisms as all of these other pathogens, which means that if we find something in Bacillus subtilis, it could potentially be conserved in a gram-positive pathogen, and we can apply that knowledge to work with these pathogens as well. So what we showed initially was that Bacillus subtilis demonstrates persister characteristics. Here, Bacillus subtilis was treated with a cell wall targeting antibiotic. So you see the characteristic biphasic kill curve here, excuse me, where a majority of the cells are dying in the first couple of hours, followed by a plateau where the survivors are just surviving, they're not dying or anything like that. And when you take one of the colonies that are formed in the plates at this 5th hour time point and restart the culture for the next day, you see that the day 1 survivor exhibits a similar biphasic kill curve characteristic, indicating that these cells are not inheriting the ability to survive the cell wall targeting antibiotic. And when you treat these bacteria, the Bacillus subtilis, with increasing concentrations of the cell wall targeting antibiotic, we see very similar kill curve characteristics, the same biphasic kill curve. So this demonstrates that the persistence is non-heritable, and it's not inherited and not affected by antibiotic concentration. So while at doing my PhD, the lab I joined, Jade Wang's lab, we are interested in the stress-inducible molecule ppGpp. And so when bacteria experience stress, such as antibiotic stress, amino acid stress, carbon phosphate starvation, what happens is that cells can combine GDP or GTP with phosphates from ATP, which is the energetic currency of the cell, to form a molecule called ppGpp, which you see here. And in Bacillus subtilis, they use 3 enzymes to catalyze this reaction: REL-A, YJBM, and YWAC. And so this molecule goes on to help the bacteria survive stress. And so what's important to note for the next couple of slides is that I'll talk about a wild type strain of Bacillus subtilis, which has all 3 of these enzymes intact and can make ppGpp. And then I'll also talk about a strain called ppGpp0 or P0 that has mutations or deletions of these enzymes and cannot make ppGpp. And so by comparing the 2 strains, we can determine how ppGpp can protect Bacillus subtilis against antibiotics. So first, we demonstrated that ppGpp does not alter antibiotic resistance. What you see here is the wild type strain and P0 strain treated with 2 different types of antibiotics, ciprofloxacin and vancomycin. And using the MIC assay, we determined that they have the same MIC for ciprofloxacin and vancomycin. If ppGpp increases resistance, what we would see was that the wild type strain would have growth or this gray color of growth in these wells here, but we don't see that when compared to the ppGpp0 strain. Next, we demonstrated that ppGpp promotes persister formation in Bacillus subtilis. So what you see here is the wild type strain treated with a cell wall targeting antibiotic. And again, we see the biphasic kill curve. Next, we treat the strain that cannot make any ppGpp, and we see a very similar biphasic kill curve as well, except that the number of survivors is 10 times less than the wild type strain. Finally, if you recall, ppGpp is formed when bacteria are stressed. And so what we did was we took the wild type strain, stressed it ahead of time, and then treated it with antibiotic so that ppGpp was present prior to treating it with antibiotic. And what we see here is that the bacteria, the wild type strain with ppGpp present does not die, but also doesn't really change in the number of colonies over time either. So it remains at around 100% or so, indicating that ppGpp can protect these cells against antibiotics. So I don't really have time to finish talking about my PhD research, but I want to leave you with several important points. ppGpp is important for antibiotic survival. It helps to create spontaneous persisters. So these are random cells in the population that have high ppGpp and this helps to protect the cells against antibiotics in general. ppGpp also helps to create adaptive persisters. So the bacteria can respond to antibiotics by making ppGpp and promoting survival. And why persistence is so important is that continued survival in antibiotics helps to essentially help the bacteria to buy time so that the bacteria can develop resistance to the antibiotic. And so I'll leave you with a couple of future of antibiotic therapy points here. So I think what's important to note is that we need to continue doing research, basic research to determine how ribosomes or how DNA is replicated is very important so that we can help to identify targets for antibiotics. Another important point is natural products. And so natural products are chemicals or compounds that are made by molds, plants, bacteria that can have antibacterial properties. So what people can do is figure out how to make these molds secrete these compounds, or how to clone these genes into another organism to make these compounds. And then we can test all of these compounds to see if they have antibacterial properties against pathogens. And finally, medicinal chemistry is also very important. So here, if you remember, is a molecule of penicillin, but medicinal chemistry and chemists are able to alter the side chains or functional groups of these compounds so that it can help to go into the cell better, or maybe it's less likely for bacteria to create mutations because of these functional groups. And with that, I'd like to talk about my path to science. Again, I'm from Somerville, Massachusetts, and I went to Somerville High School. And while I was there, I was lucky enough to participate in an outreach program that was hosted by Biogen IDEC. So at this outreach program, I learned a lot about biotechnology techniques and careers in biotechnology. And I was also given the opportunity to work with a mentor to develop my own project. So that was a really great experience I had. At Wellesley College, I majored in biological chemistry and joined 2 labs. So the project I worked on was a collaboration between 2 labs. So it was collaboration with the biological sciences and also with chemistry. And so I had a lot of fun working on cyanobacteria and its ability to tolerate acid stress. I was also given the opportunity to present my work at the American Society for Microbiology. This is me with my poster. After graduating from college, I worked at a small biotech company called Rapid Microbiosystems as a research assistant. And after 2.5 years or so, I applied to a PhD program. I applied to the microbiology doctoral training program at UW-Madison. And this is the lab I joined, Jade Wong's lab. And this is my boss or principal investigator, Jade Wong. And we worked on ppGpp and a lot of other projects as well. During my time there, I was also given the opportunity to talk about my research again at American Society for Microbiology, this time as a guest speaker. And research is really fun, and it's a lot of hard work. So there were times that we were goofy as well. So the program that I was in every Halloween hosted a party where it was themed. And so here is a picture of myself in the lab dressed up as the Magic School Bus, where we were the bee episode, and I am the bus right here. And then finally, after graduating, I actually got a job again at Rapid Microbiosystems, this time as a scientist. And so Rapid Microbiosystems manufactures the Growth Direct, which you can see here, which quickly detects microbial contamination in pharmaceutical products. So needless to say, STEM plays an important role in the pharmaceutical industries. There are a lot of people there with microbiology background, and they work in several types of departments. So research and development, which is the department I work in, quality control and applications, and then also engineering are spread out in research and development, quality control, and manufacturing. So all of these departments and disciplines work together to develop new products. And my role as a scientist is to ask questions, design experiments to address these questions, act as a subject matter expert, and mentor and train young scientists. And with that, I'd like to thank the Headwaters Science Institute, UW-Madison, and my own company. And I'd be happy to take any questions that you might have.
Thank you, Jessica. That was awesome.
Thank you.
I love that you finished kind of with how you got into your career and what a scientist like you actually does by day. That was fascinating.
Sure, thanks.
So to kick us off, I want to start kind of where you ended and just ask if there are young scientists watching who may want to go into the field of science, maybe microbiology, but aren't quite sure where they want to go. How did you identify what it was that you wanted to study?
So I think I've always been interested in the sciences really early on. And I think my experience at Biogen IDEC had really helped me to really enjoy looking at things on a molecular level. And so I think learning about what kind of options are out there and asking people questions, you know, if people are going off to college and you research the types of scientific research that's available at the college, and then make appointments with the professors, or if there's grad students or undergrads, to talk to them about their research. You know, it might seem scary, but people love talking about their research. They will go nonstop. So just having that conversation to figure out if it's something for you. And if there's summer research programs too, you can see like, oh, would I be interested in bench science, right? Or is it something I'm still interested in science, in the sciences, but not necessarily working at a bench top. Mm-hmm. And so there are ways to figure that out. And I think just asking questions in various departments, colleges will definitely help to narrow it down.
That makes sense. And it sounds like you're saying having the experience will quickly tell you whether you want to go in that direction or not.
Definitely, yeah. As a grad student, we have a lot of undergrads that want to be part of the lab, right? Just to experience what research is like, you know, attending lab meetings and presenting your research. And it's definitely not for everybody. And so figuring that out by doing a summer research program will really help you figure out whether you want to do that or not.
Mm-hmm. Mm-hmm. So that leads us into Spencer's question here, specifically for high school students interested in the opportunity to work or intern in a lab. Do you have suggestions for how they could find those opportunities and how to make the most of it?
Yeah, I think so. I feel like I was pretty lucky in that, you know, Somerville is very close to Cambridge, where Biogen IDEC is located. So, you know, they want to do an outreach program. I think, you know, there are biotech companies or other science institutions that are always interested in outreach. And so, you know, maybe contacting the company itself to see if they have opportunities like that open, or even just asking if they can shadow someone during the day, right? That'll be very helpful for you. Mm-hmm. And then even at local colleges, you know, being able to talk to some of the grad students or professors and maybe getting some hands-on work there, that could be helpful as well. They're always looking for volunteers, you know, so just ask.
Great. Thank you for the recommendation. So we're going to get into some questions that kind of switch gears here. This question is from Laura. Are there any diseases that might revive as the bacteria have become resistant?
Diseases that might revive as they become resistant. Well, so I think, I'm not sure. I think, you know, now that there are antibiotic resistant bacteria that's kind of lurking around, those are bacterial infections that are getting harder and harder to treat. So in a sense, those, I guess, have revived. But other than that, I'm not sure that there are others that would revive.
Okay. And have people's immune systems weakened since we all have been taking antibiotics and we have these modern cures for almost every illness?
I don't really think the immune systems have weakened. I think, say, there are things that people can do to help with this antibiotic crisis. So one thing is that when you're prescribed antibiotics and you're prescribed it for 7 days, but you only take it for 3 days and you start to feel better, you should still keep taking it for the full 7 days, just so that you keep on suppressing the bacteria, and that it actually gives your immune system a chance to help fight the disease as well. I don't necessarily think the immune systems have weakened, mainly because it still needs to work with the immune system to combat these bacterial infections.
Gotcha. Another question about bacteria. Are there certain kinds of bacteria that are more likely to become resistant?
So that's a great question. A lot of bacteria, they are able to acquire plasmids, or they all have the potential to mutate. And it's down to whether they have that machinery inside of the cell that can actually help them increase mutations or not, and whether they can actually acquire these plasmids or not. So it's really down to the bacteria. And bacteria, they find so many different ways to survive, right? Like make spores, develop persistence, you name it. So there are definitely ways.
And are you saying then, to address the second half of this question, is there something in their genetic makeup specifically?
So there are definitely error-prone machinery that can help to mutate DNA. So I think certain types of bacteria have that.
So I'm wondering, when you were talking about persistence, you talked so much about that curve.
Yes.
And we saw the downward slope, and then we saw kind of an evening out at the bottom. What is happening at that point once the presence of bacteria evens out?
That's a great question. So a lot of times what we've shown is that these bacteria are just kind of dormant. They're not growing or living, or they are living, but they're not growing or actively dividing. And a lot of the antibiotics, what they do is focus on machinery that the bacteria need to grow, right? So if they're not actively growing or dividing, then they can kind of just hang out and not die in the presence of the antibiotic. But there are also other types of mechanisms that probably work all together in order to protect the cell. So ppGpp is one of the mechanisms, but there are certainly others as well.
Interesting. So an overarching question here: what is your favorite part about the research that you do?
Currently, or at UW-Madison?
Currently.
Yeah, so right now I like learning about how to improve a product. That's not something that I've done before, right? So learning how to improve a product is very interesting. Working with all the different groups and disciplines is also very interesting as well. We're microbiologists, but so a lot of times we don't really think the same way as a lot of the engineers, and then they don't think the same way as us either. So being able to explain those concepts efficiently is very important. And there's always challenges, but they're interesting and good challenges.
I have kind of a follow-up question to that. I think we've heard from a lot of lab scientists who do very intricate work that can seem very detailed to the outside person or the outside eye. Do you have to work to make your studies relatable to the general public or to other people that you have to work with?
Oh yeah, definitely. We need to be able to talk about how we do the assay and why the experiment and why it's important, so that say people in other departments like marketing, right? Like they need to understand the science, but also why you're doing it too, but not necessarily the detail, right? So it's very important to be able to communicate that effectively.
Mm-hmm. Absolutely. So that kind of leads me to the wrap up here. The point of Lunch with a Scientist is to get scientists like Jessica Tse Barra to communicate what they do and to help you understand why science is important. So Jessica, thank you so much for being here and for being a part of that.
Thank you for having me, and thank you to Headwaters Science Institute.
So if you're watching, if you like what we're doing, please share this with a young person who you think might be inspired. If you want to help us continue to keep these programs free, you can go to headwatersscienceinstitute.org/donate. And Jessica, I hope you have a wonderful day.
Thank you. Bye.
Bye.